Yes, so thank you, Erwin, for this opportunity at a spectacular place. Never been here. So it seems like the thing to do this morning is to give your own definition of active matter. And so why not? That's my definition of active matter, which simply says that active matter converts scalar energy into a vectorial output. The scalar energy would be a chemical reaction. The vectorial output can be a chemical gradient, ion pump that you see in cells. It can be directed motion. It can be swarming. And of course, this violates Curie principle. So what I'm going to do today is to show examples of this from molecular scale, which is what, 5 to 10 nanometers, to maybe about 100 nanometer particles. So all of these would be chemically powered. And the starting point are enzymes, which are, of course, the master catalyst. And one of the things that we observed early on was that when enzymes catalyze reactions, they generate a propulsive force which causes them to enhance their diffusion. So this is just one example where catalase converts hydrogen peroxide into water and oxygen. And as you increase the substrate concentration, which is the hydrogen peroxide concentration, the diffusion goes up of the enzyme. If you inhibit the catalase, in this case it's sodium cyanide, you get the base diffusion. Now, why? No one knows for sure. And there is controversy, as some of you might know, that what is the extent of this enhancement in diffusion? Is it 5%, 30%, or what is it? What is known and not disputed is that if you put these same enzymes on particles and activate them by adding a substrate, the diffusion of the particle will also increase. But that's a different mechanism. That's more of a diffusiophoretic mechanism, which is not possible for a single enzyme because they spin so fast that they cannot detect their own gradient. But anyway, to us, what is much more interesting is the fact that enzymes undergo chemotaxis. Now, chemotaxis is basically things moving towards food or fuel or whatever you want to call it. This was thought at one point, I don't know why this got screwed up, but anyway, this was thought at one point to be a hallmark of living systems, but of course, stupid enzymes do it. And you can show it in many different ways. Here's just one example of a 3-inlet microchannel. And you put equal concentration of the enzyme urease in all 3 channels. And then if you pass its substrate through the middle channel, the enzymes from the side channels will focus in the middle channel in search of fuel. So you go from an equilibrium distribution of enzyme to a non-equilibrium distribution. How does this happen? Well, the simplest case is when you have an irreversible reaction. In this case, if you have a gradient of substrate, of course, the enzyme will bind to the substrate. And where the substrate gradient is higher, there'll be more substrate-bound enzyme, and where the substrate concentration is lower, there'll be less substrate-bound enzyme. And then it depends on which one diffuses fast, the bound enzyme or the unbound enzyme. And whichever diffuses fast, the net movement would be in that direction. So that's simple. It depends on the difference in diffusivity of the bound versus unbound enzyme. Okay, so that's what it is. But of course, most reactions are not irreversible. And so in that case, so for example, this is the usual enzymatic reaction where the substrate binds to the enzyme and then the product is released. I call it L because it could be substrate or product, doesn't matter, but it's bound. And in this case, it's a two-step reaction, and there's an activation energy associated with both steps. And you can then talk about the kinetic asymmetry, which is the difference in activation energy between one step and the other step. So this is the kinetic asymmetry of this particular reaction. And if you take this into consideration, what you find is that the direction of chemotaxis now not only depends on the difference in diffusion between the bound and the unbound enzyme, but also on the kinetic asymmetry here shown as the difference in off rates, product coming off versus the substrate coming off. And if either one of them is 0, there is no chemotaxis. And then the direction depends on the relative weight of each of these and the relative sign. So I won't go through the whole derivation here. So is this relevant? Well, so we all have these enzyme cascades in our body. For example, there is a glycolysis cascade. And so the food you eat eventually gets converted into ATP through this glycolysis cascade, which involves some 20-odd enzymes. What basically happens is the first enzyme makes a product, which is a substrate for the second. Second makes a product, which is a substrate for the third, and so on. And what people have found is if you provide the substrate for the first enzyme, all the enzymes in that cascade come together. This is known as a metabolon. You can see this. This is a different cascade. This is a purine synthesis cascade. And you can see that they come together. In fact, the scale bar is 10 micron. It's easy to see. When the substrate is used up, they disperse again. And there's a reason why metabolon formation is advantageous, because if you're just going to go handing over from one enzyme to the other, if they come together, it's much more efficient. You don't lose the products in the bulk, et cetera, et cetera. But the fundamental question is, what makes them come together? And our hypothesis is that it's all chemotaxis. So the first enzyme makes a product which is substrate for the second. So the second enzyme then moves towards the first because that's where its food is. And then it makes a product which is the food for the third enzyme. So the third moves toward the second. And in this way, all of them come together. Well, is this true? So we looked at the first 4 enzymes of the glycolysis cascade, hexokinase. I'm not even an enzyme chemist. I can't even pronounce these guys. Anyway, hexokinase, isomerase, another kinase, and then aldolase. You provide it with D-glucose, which is the usual glucose. Hexokinase converts it to this, which is the substrate for that, which converts to this, which is the substrate for the next one, and so on. And we labeled the first and the fourth enzyme. And we used, again, a microfluidic device where we introduced the first and the second enzyme in one flanking channel, the third and the fourth enzyme in the other flanking channel, and then you pass D-glucose. And now what's going to happen? Well, hexokinase would immediately chemotax because its substrate is D-glucose. This guy has to wait until its substrate is produced. and so on. And then aldolase has to wait until the first 3 enzymes have done their job. And so there's going to be a time delay between the chemotaxis of hexokinase and aldolase. And that's exactly what you see. So it seems like this is a mechanism for these metabolin formations in your body. So this is actually an example of how information can arise out of energy or chemical gradients. So if I have 1,000 proteins in solution, if I add a substrate, only those enzymes which are part of a cascade will come together, and they will come together in a temporal manner. The second thing I want to point out is that this enzyme is following its substrate, this enzyme is following its. So this is moving this way, and this is moving towards it first. It's an example of non-reciprocal interaction, which Hartmut talked about. And it's an apparent violation of Newton's third law, which he also talked about. And I'll get back to this in a minute. So chemical gradient is information. It is a vector which has a direction and a magnitude, and arguably it's the first source of information in the prebiotic world, which got molecules to assemble. We were assembling enzymes to make metabolins. Chemical gradients can cause molecular scale assembly. And so this allows you to make swarm-based functional assembly, and I'll talk about that next. So, well, there's a lot of work, as Hartmut pointed out, in non-reciprocal interactions. We were wondering, is there a mechanism by which molecular scale systems can have non-reciprocal interaction? So we constructed this, which is you have a protein, and a kinase will phosphorylate the protein, and then a phosphatase will dephosphorylate it. And so you go round and round. You can, of course, have larger metabolic cycles and so on. And to address this, we said, OK, so we have the kinase immobilized, and the phosphatase in solution. You have a semi-permeable membrane, which allows things to go in and out. Or you can do the other way around. And basically, what we're doing is looking at what phosphatase does and from the point of view of kinase, or the other way around. And if you do this, you see that simply diffusion and kinetic asymmetry is sufficient to cause non-reciprocal interactions. You can, in fact, calculate a chemotactic velocity, or come up with an expression for chemotactic velocity. And if one of them is different for the kinase versus the phosphatase, you will have non-reciprocal interaction. So non-reciprocal interaction comes about from at the molecular level. Again, it's a source of information at the prebiotic world. And one of the interesting things about non-reciprocal interaction is that you get giant fluctuations. So if it's attractive interaction, two things come together, and that's the end of that. If it's a repulsive interaction, there's a little bit of fluctuation. But if it's non-reciprocal interaction, you get giant fluctuations. And others have speculated, Ramin, for example, that these fluctuations allow these things to come together in primitive metabolic cycles. So kinetic and diffusion asymmetry is sufficient for enzyme chemotaxis. And it is also the root of non-reciprocal interactions, which can have large fluctuations, and it allows dynamic control over self-assembled structures. What about particles? Can you also see same kind of interaction with particles? So again, so if you attach enzymes to particles, and then this particle, this is its substrate, will move this way, produce through the reaction a different chemical, which will be the substrate for the second enzyme, which will then move this way. So this is a non-reciprocal interaction. To do this, and it takes longer than you can do it in microfluidic channels, which is 30, 40 seconds. Here you need several minutes. So what we do is we have a hydrogel inlet, and we have the substrate come in through here, and you put the enzyme-attached particles here, and you see what happens. And so we have two kinds of particles. The first particle has acid phosphatase on it, and the second particle has glucose oxidase on it. And so what do you expect? And you're going to have introduced glucose 6-phosphate, which is the substrate for the first particle. And so it will chemotax towards the source. And in the process, it will form D-glucose through the reaction of the glucose 6-phosphate, which is the substrate for the second set of particles. So they will follow. So again, it's a predator-prey situation, one population of particle chasing the other set of particles. And so you can see this. So this is the acid phosphatase velocity towards the source. This is the glucose oxidase velocity that's chasing the acid phosphatase particles. If you use a different substrate that does not produce glucose, then only the acid phosphatase moves, the glucose oxidase doesn't move, which is the control experiment, if you will. So what that allows is distributed functionality. So you can think about two sets of particles, two populations of particles. Let's call the first particle the seeker particle. So what does it do? It hangs around until it finds a source, and then it moves to the source. So it moves up, chemotaxis up the gradient towards the source, and it secretes a second chemical, which is the substrate for the second set of particles. These are the, let's call it the delivery units. And so they will hang around until they see the signal from the first set of particles, and they will move in. And they can show where your source is. They can deliver a drug or whatever you want. So you can have these populations of particles communicating with each other, doing things cooperatively. And so now there's a lot of work. People have been making vesicles and so on, putting in enzymes and loading it up with all kinds of things and using chemotaxis to direct them. You can do positive chemotaxis. A good example is this paper from Battaglia's group, and Giorgio Volpi was part of it. It turns out, thank God, that our brain has more glucose than our blood. And so if you use particles that are functionalized with glucose oxidase, for which glucose is a substrate, they will cross the blood-brain barrier in looking for glucose. Is it agency? I don't know. Okay.
Is that Sean?
That's the paper. That's the paper. Ask him. Don't ask me. We've shown, in other contexts, we've shown both positive and negative chemotaxis. Well, I was all good with the talk, and then Erwin said, show me something that happens in a cell. Okay, so I had to put another slide in. So this is chemotaxis happening inside a cell. So mitochondria. The function of every cell has mitochondria. The function of mitochondria is to produce ATP. Okay? And so it secretes ATP. This ATP goes to the rest of the cell, and some of it is secreted outside the cell. So we then took 100 nanometer particles, functionalized them with ATPase enzyme, for which ATP is the substrate. And then you put them outside the cell, and we use human endothelial cells. So what this shows, so the NP ATPase are the particles functionalized with ATPase, and these guys are not functionalized with ATPase, or they're functionalized with ATPase, which we have killed. So this is just outside the cell, at a height of 5 microns from above the cell. And you can see that the ATPase particles, this is a blue, starts to collect near this cell. The ones that are not functionalized are distributed evenly throughout the solution. So the ATPase particles come close to the cell, they get endocytosis, goes into the cell, and then they collect near the mitochondria. So the red is the mitochondria, it looks blue here, but anyway, this white-blue dots are these particles. And you can see that when you don't have ATPase attached to the particles, some of them are taken up, but they're not particularly localized. These are. This is a mitochondria, and it's localized around that. So it happens inside cells. Okay, the last thing. I got time. So what happens if you prevent these enzymes from moving and you attach them? Okay, they're generating an impulsive force. This force gets transferred to the fluid. So they act as miniature fluidic pumps. They pump fluid. It doesn't matter how you attach the enzymes. You can do it electrostatically, you can do it covalently. You attach the enzyme, give it food, and you can put tracer particles to follow the fluid flow. And if it's a confined chamber, then you get a convective flow. Depending on the enzyme, you go this way or this way. And it's, again, very general. And just like enzyme diffusion, it's a function of the reaction rate or substrate concentration. You can see these particles moving. So I just show you 3 enzymes. And you get decent velocities, several microns per second fluid flow. Eventually, they will stop because they're eating up their substrate. So they will stop. So, they run down in a few hours or more. But if you add more substrate, you can recover the pumping velocity. Okay, so what's the mechanism for this one? Well, the mechanism for this is density driven. So if the products are lighter than the reactant, they're going to rise. And so the fluid, because of fluid continuity, they will come in from the side. We call this inward pump because the flow is inward near the enzyme patch. If the product is denser, they spread out and they come in. So this is an out pump. Now you might say, how on earth do you know any of this? Well, So you can take these pumps, right? And you can invert them. So now think about what's going to happen. So this is your upright pump. And let's say the reactants are lighter than the product. And so they're going to rise. And so the flow will be inward here and outward on the top. Now if you invert it, now the lighter products are not going to go down. Of course not. Because of gravity, they'll spread out, and the flow will reverse. And this is the only mechanism where the flow reverses. If it is a diffusiophoretic mechanism, there should not be a reversal of flow if you just turn it upside down. The other thing I should point out, that if you measure the velocity as a function of the height, at the surface is going to be 0, no slip boundary condition. About 25% height will be the maximum velocity. Then at 50% height, it's switching direction. Velocity goes back to 0. 75% height, it goes maximum. And then right at the top, again, no slip boundary. So you get an S-shaped curve if you measure the velocity as a function of the height. Okay, so these are really pump sensors, because they only pump when they sense their own substrate. So they can sense and pump. So one of the problems with trying to publish anything in Nature X is you can't speculate. So we submitted this paper. We said, so this shows that you can deliver drugs on demand. And of course, the editor said, show me. So we took a hydrogel, and we put glucose oxidase on it, and we filled it up with insulin. And you can put it in a solution of glucose. Now remember, the pumping velocity depends on the concentration of your substrate. So higher the glucose concentration in the surrounding, more is the pumping, and more insulin is released. And so the insulin release depends on how much glucose you have in the surrounding. So this is enzymes on hard surfaces. What about enzymes on soft surfaces like a membrane, right? Our cells, in fact, most of the enzymes in our body are membrane-bound enzymes. And they're often targets for drugs and viruses. COVID virus targets one of these. And there's a lot known about how curvature changes enzyme activity. Not much is understood about how enzymes change the curvature and other membrane properties. I don't think I have the time to go through it. All I'm going to say is it's fluid flow, because you can attach the enzymes on the membrane, you can add particles, and you see the particles moving. And depending on what enzyme you attach, they move towards or away from it. You see the S-shaped curve. That is a telltale sign of pumping. So yeah, you see active fluid flow on membranes. And they can possibly control cellular dynamics, tells you about how to control in confined 2D environment. So this seems relevant. So Irwin suggested yesterday that one should be argumentative. So Arvind, this is your paper, okay? And you posit that you can have non-protein driven, well, actually it was Petrashvili who did the experiments, can cause transport on membranes. And suggested that it was a diffusiophoretic mechanism. I have no idea what the diffusive mechanism here is, but I would like to suggest that it's fluid pumping, which is causing the transport. And the easiest way to find that out is you put some particles on the surface and look at the velocity, horizontal velocity as a function of the height. This is an interesting one. Christine Jacobs-Wagner showed that if you take a cell, and in her case it was an E. coli cell, and you stop all the enzymes in the cell becomes glass-like. And it is only when you turn on the enzymes that all this churning and motion that causes the cell to fluidize. And so it seems like this is all quite relevant. Okay. I think my time is almost up. I got a few minutes. So that was what I wanted to tell you, but every decent meal should have a dessert at the end. So this is my dessert. Okay, I wanted to show you how powerful these enzymes are. So now we're talking things that are centimeters in size, okay? So these are real macro things. So I can take a sheet of plastic, I can half coat it with gold, attach enzymes to it. Okay, so I can make a windmill, for example, and this is unsymmetrically attached, as you can see, and I can take this with the enzymes attached here, put it down like this, add substrate to the liquid, it should turn. Okay? Now, suppose you put two different enzymes on these yellow patches. One that pumps in one direction, and the other that pumps in the other direction. Okay? Then the rotation will depend on whose substrate you put in solution. Okay? So these are the windmills. This is the gold-coated surface. You can't see the plastic that's not coated, so it's this really, but you can't see it. Anyway, trust me. So if you add the substrate, so I have both phosphatase and urease on it. So if I add the substrate for the phosphatase, okay, it goes anti-clockwise. And if I add the substrate for the urease, it goes the other way. Okay? So these enzymes are moving centimeter-sized objects. Okay. So I think I've told you that enzymes can generate force to move large particles. The movement becomes directional chemotaxis. I didn't talk about this, but if you also add inert particles to an active enzyme bath, the diffusion of the inert particles will also increase. They can act as pumps. So it's clear that the enzymes can provide sufficient force for stochastic motion of cell membrane, cytoplasm, organization of metabolons, and so on, so forth. So it looks like it's a new area of mechanobiology, the intrinsic force generation by enzymes and their role in biochemical regulation of cell function. So let me acknowledge the students and postdocs who did this, my collaborators inside Penn State and outside Penn State, people who funded us. Hasn't come out yet. I just checked before the talk. But if you wanted to know where things are going, this was supposed to come out today at some point. Okay, this is Germany. I need to show you something from the Audi Motor Company. This is taken from an Audi magazine, and it kind of summarizes what we're trying to do. A nanoscale submarine moving through a human capillary. What's going to be the fuel? Well, it's going to be glucose, because your blood has a lot of glucose. And so you would power the submarine using glucose oxidase. Where are we now? Here. This is the first computer mouse 60 years ago. So, in another 60 years, we'll get back to here. All right, Igor, I'm done.
Thank you.